Alright, welcome everyone to the second lecture of the experimental design lecture series. And today, after having reviewed last time all the different principles on which you should base your research and your experimental design, we're going to have a look at a case study. And in this case, we're going to look at the target for quinine. And I'll give an introduction to what that means and everything as well in a few minutes. And the subtitle of this lecture is Everyone Can Be a Good Scientist. And I completely believe that because I think that regardless of whatever you have access to or whatever capabilities you think you might have, I think if you follow good research principles, you can contribute a lot. And it's quite important to actually learn it from someone. Usually you learn it from mentoring in different labs where you work. So starting off, I'm always going to have this little bullet point of different things that you basically will do during your research and that are part of the scientific method. In the top right corner, and I have them numbered, and basically I annotate different bullet points in my presentation so you know how it relates to the things we talked about last lecture. So I'm not going to specifically point them out, but they're there for future reference if you want to go back and check this again. So starting off, what's quinine? Quinine is an anti-malarial drug, and the structure is the one you see there. And there have also been structures that are related that have been made that are synthetic, and this is mefloquine, for example. It's also an anti-malaria, but this one is a synthetic compound. I think it was developed by the US Army at some point. And you can see the structures are quite similar. The only reason I bring it up is because the studies that we're going to see also involve the use of mefloquine as an analog to check whether it has any relation to quinine because of their similar structure. This general quick tip, you'll know if something's natural or not if it has a trifluoromethyl group like this structure does. As far as I know, there are no known chemical compounds that are naturally occurring that contain a trifluoromethyl group. So this is an entirely man-made substance as far as I know. That's just a tangent. But anyway, this quinine has been isolated from the cinchona plants all the way back in the 1800s. And these kinds of extracts from plants have been used traditionally to treat malaria. And this has been done for very many years. I think even as early as the 1600s, they have records of people using extracts from these plants to treat malaria. And you don't get one compound when you use extracts from plants like this. You get a whole family of compounds that are structurally related. And here you'll see where I have R1, R2. Can I use that as a laser pointer instead? Yeah, there we go. So you'll see here R1 and R2. These just denote that there's different kinds of substitutions on the structure there. And this is why you get a family of related compounds. And these compounds are actually quite famous in chemistry because historically their synthesis was quite a big deal in terms of organic chemistry. They came in the late 40s or so from a very famous organic chemist called R.B. Woodward, and he was at Harvard at the time. And they also used synthesis back then as a tool of understanding what the actual structure of the compounds were because you didn't have the tools that we do today. But anyway, this is a bit of a tangent. The main point of this is the mechanism of action. So what exactly this drug does to combat malaria is not known. So this is quite a big deal, right? This has been used for hundreds of years and nobody knows how it works. So the question that's going to be investigated here, thank you. So the story that's going to be investigated here is what is the mechanism of action for the anti-malarial properties that we see in quinine and presumably structurally related compounds. So you'll see why I say that in a bit. But anyway, the hypothesis that they start with, which is implicit, which isn't stated outright, is that quinine targets a protein. So typically drugs, with some exceptions, will just simply target a protein and inhibit it or target an ion channel or something like this. And the other implicit assumption or hypothesis rather is that the protein is found in the Plasmodium parasite, which is the parasite that causes malaria. Alternatively, the protein could also be found in the cell it infects, but this wouldn't make too much sense. Otherwise, it might also might have effects on people that don't have malaria, but this is treated later in the paper. So how do you actually figure this out? This is a super unspecific question since you don't really have a hypothesis of which protein it's targeting, right? You need a method that's going to screen for a lot of proteins. So it screens proteins, and it ideally would do it in cells because then you're looking at the sort of model system that's closest to what you see the effect in. And you'd want it to be high throughput because, as we said, we don't know what we're looking for, so you need to check a lot of proteins. So what the authors did by looking through the literature, which comes, as I said earlier, I've annotated each of these points, and this comes back with prior knowledge when you look in the literature, they identified a technique called a cellular thermal shift assay. And practically this bases itself on the fundamental, observation that drug binding to a protein increases its melting point. And why is that? Well, it's not particularly complicated, because once you get binding, essentially the energy of the whole complex is lower. This is why you get binding. This is what drives binding. And a complex which is overall more stable needs more heat to denature and destroy the tertiary structure. So when you bind something to your protein, you're going to have a higher melting point for the protein compared to when it doesn't have anything bound to it. Another thing is that when proteins melt and they denature and they no longer have any of their structural properties or anything like that, they tend to precipitate out of solution. Whereas if they're stable, all the right things are pointing out in the solvent, so they're soluble. And then the last thing, this is what they actually did different to the already existing technique, is they coupled this already known assay that looks for precipitation of the different proteins at different temperatures in the live cells directly. And they coupled this to multiplex quantitative mass spectrometry. I mean, this is just a fancy word. It means a lot of things and it has a very specific meaning. But overall, all you need to know for the purposes of this lecture is that essentially it allows you to look into a protein mixture and identify what proteins are there. This is the take-home message. Basically, this lets you identify what proteins you have in a given mixture. Quick overview, I mean, I'll go over this again in the next slide because it's a bit complicated, but just a quick overview is you take your cells, you heat them up, proteins precipitate, at least the ones that melt at that temperature. You do centrifugation and cell lysis, so you basically spin the sample really fast, so all the stuff that's precipitated will go to the bottom of your tube. And cell lysis is, you literally make your cell explode. And then the original method would use quantification at different temperatures. It would do quantification of the drug at different temperatures using Western blot, but they did it with MS, so this is their addition to the new method, right? Just keep that in mind. So previous work showed that the target is in the cytosol, so this is not something they need to worry about because this method is not really suited for membrane proteins. But knowing some previous knowledge about how quinine works and where the target might be lets them identify what method is suitable, right? And here, every slide, once I go through what they've done, I'm going to try and provide a key point of what the actual authors did in this particular example, and also what lesson you can take away from what they did and apply to your own research. So in this case, the authors read very carefully the literature, identified their needs for their experiments, and found a suitable method to assay what they're looking for. And what lesson can you take away is a saying I was taught a few years ago, and I really like it, is that a week in the library can save a month in the lab. So if you do your homework properly, it could end up that you actually save a lot of time in the lab from doing stuff that isn't particularly productive. So now, moving on to the actual experiments they did. So they had a plan of doing 4 different things. So one, they're going to do what's called a drug response CESTA, which is the assay we talked about in the previous slide. So you take a set temperature and you give the cells different concentrations of the drug. Second one they want to do is you take the same concentration of the drug and give it to the cells, and you heat them up to different temperatures. Then there's 2 different setups of the types of cells they're using. One is you take the whole blood cells with the parasite in them, and you do your experiments on this. And then the other one is you take the same culture, but you essentially explode everything and take anything that's soluble. So this is called cell lysates. So they have 4 different approaches. So 2 that have to do with temperature or drug response, and 2 that have to do with the setup of their cells. And you'll see why that's important later. It's a very good experimental design in that they overlapped various scenarios by doing this. So the reason they set up the 2 different cell experiments is that it can also target 2 stages of parasite life cycle, so when they're inside the cells or when they've matured enough to go outside their cells. The dose response and temperature response basically check each other, so if you get a weird result in one and you don't get it in the other, it's possible that you should start looking at other things, and there's an example of this later again, so we'll review why this is important. And the fact that they have these 2 setups again, where you have the cells intact, ensures that the cell lysis is not important to the result you get, as you have one setup where the lysis is done after the heating up in the drug treatment and one where it's done before. So these should be the same for stuff that's real results. So there's an implicit assumption also in the method they're using, and it is that changes in the thermal stability are only due to drug binding. So I'll explain exactly what I mean by that. This is not a safe assumption because there are other things that can change the thermal stability of a protein, which can be protein-protein interactions, a whole host of things that can happen in the cell that are actually a result of drug binding, but indirectly. So it's not that your drug is binding to this protein and changing it. It's rather that your drug is binding to a protein. This is causing a change in a metabolic pathway or a signaling pathway or something, and this also changes another protein further down the line. And this is why they have these 4 different setups. Because there are other effects that can change the melting temperature, you need to check it in multiple ways to make sure that the effects you're seeing are actually because of what you think they are, right? So here's just a figure straight from the paper that sort of summarizes exactly what they did. So they have, the same kind of culture, and in one of them, they take out the cells and the parasites, they do lysis, and then they have a sample. And in the other one, this is not so important. They just remove some protein so they don't have so much background. But basically here, they just take the cells directly and then do their experiments. And the experiment is you split your samples, and then you either add different concentrations of the drug, which is this experiment we talked about, or they just don't add drug or do add drug at the different temperatures. Following that, they just take the soluble proteins. Basically, this is just treating it appropriately so that you can do the mass spectrum analysis to it. So the simplified version is they take the cells, either they break them up or not, they add drugs or temperatures as they want, and then they figure out what's in the soluble fraction. So what's the key point here? The authors identified the assumptions of the method, so this is one good example where other effects can change the melting point. And they put validation checks in their experimental design. The validation checks that we're talking about are these 4 different experiments. So despite the fact that all these should give you the same answer, you're still doing it 4 times in 4 different ways to validate that what you're seeing is real. And what lesson can you take from this is identify and evaluate any implicit assumptions of your methods. So things that your method assumes you know, but you maybe haven't thought about. So you really need to check. Also, the people who develop the methods ideally should tell you what these are, but you could maybe ask someone if you don't know as well. So first, what they do is they take their cells and they simply apply their method to see what proteins are there, because they need to know the background. So if you're looking for change in something, obviously you need to know the initial state. So this is the first thing they do. So they just do the different melting points for the 2 setups, so the lyse cells or the intact cells, just with nothing in there. Native, normal, no changes. And they see a difference. So this is why it's important, right? And this is why you have to do your control, because even though they should give the same answer, they don't give the same answer. So about 44% more proteins had a higher melting point in the lysate than they did in the intact cells. So this is why it's really important to do controls. And they figured out that probably this is because during the cell lysis, so during the point where you explode your cells, probably you damage some proteins and they're destroyed. So the same thing that would happen with heating would happen with aggregation. And basically you form stuff that would be more stable but doesn't actually exist in the cell. So knowing this, they made sure that when they're then identifying stuff in their MS analysis, they just have more stringent criteria. So I mean, I simplified it a lot with the quantitative MS, but there's a whole bunch of programming and stuff that goes into figuring out exactly what you have in there. So they fiddled with their code to be more stringent in what they see. So what's the key point here? They saw weird data. So this point where you have more proteins in one or the other, despite the fact that you're expecting the same result, this was not what they expected. And instead of just accepting, oh, that's fine, that's how it is, no, they tried to figure out why that is, and they also tried to correct their experimental design. So they corrected the code, they had it in their MS spectra in this case, in their analysis of the MS spectra. So the lesson from here is always look at your data critically. So just look at it, does it make sense? If I'm expecting the same result, did I get the same result? And if you see weird data, deal with it. Try and do something about it. Try and understand why it's there, and change your experimental method or whatever to actually address the problem, and don't just sweep it under the rug. The next thing they did is they took a drug that has a known target, because this is not what they're trying to do, right? They're just trying to sort of validate that their method works properly. They take a drug with a known target. In this case, they took a pyrimethamine, and this targets this protein, which I think is this is the name of the parasite, dihydrofolate reductase, something, whatever. Anyway, a protein that exists already and it's been characterized and they know what it targets. This is also anti-malarial and they just simply know how it works. So they run the experiment with the two different methods, as we said, right? So you have different concentrations of drug at one temperature or the same drug concentration at different temperatures, and they find exactly that. They find exactly the protein that they knew it targets is the only one that shows a significant stabilization. And they find a number that they can correlate to literatures, to known stuff. So not only do they get the right protein, they also get the right dose, roughly. This is the takeaway there. So authors, they tested their method with a known target to see that they get the right result. So you plug something in and you know what you're expecting. So before they didn't know what they were expecting, right? They knew that the two methods should give the same answer. In this case, they know what the answer should be and that the two methods should give the same answer, and they do get that. So this means that their method is working correctly. What can you take away from this is you always need a positive and a negative control for your method. Because if either of them aren't working, something's wrong and you need to stop. Okay, so then they moved on, and they knew from the previous experiment that they know how to monitor all the proteins they're looking for. This is fine. This is what they did with their controls, right? And they also know how to look for a specific protein. So if they know what they're looking for, they know they can find it. So what happens if you don't know what the protein you're looking for? So you see that what they're basically doing is they're slowly building up to the point where they've done enough experiments to be certain that the next step is reasonable, and the next step they want to get to is putting in drugs that they don't know what they're looking for either. So they want to go for something intermediary now. So they know roughly what they're looking for. In this case, they use this broad spectrum cysteine protease inhibitor. What does this mean? So in your body, you have proteins that break down other proteins, and these are called proteases. And then there's a specific class that use a specific kind of amino acid in their active site called cysteine. So they know that they're looking for proteins that are proteases and that have cysteines in their active sites. And these are generally quite well known and characterized, so you'll know if you find one, more or less. So even though they don't know what specific protein they're looking for, they know they're looking for one that acts kind of like this. So it's not quite the same as the experiment they're going to run later, where they're sticking these in and they have no idea what they're looking for, right? So they went from, I'm just looking at everything, to, I know exactly what protein I'm looking for, to, okay, I'm looking for a protein that should kind of do this. And when they do that, they found 4 proteins that have a stabilization, these ones here, and they are all cysteine proteases. So it seems like the results also still work here. This is a bit of an extra control, and honestly, I really like this. This is one of the parts that makes me nerd out a lot, because they went an extra step to have a control to validate their method, which is great because they kind of did method development, so I think that you really need to demonstrate that your method is robust when you do this stuff. But now they validated their method to an extent where you can't really think of anything they haven't done before answering the question they wanted to, right? And what's your takeaway from this is to change one variable in the experiment at every time. So as we said, they started from just looking at all the proteins, then looking at all the proteins and finding an answer they knew they wanted, then looking at the proteins and having a hint as to what they're looking for, and then they want to move over to not knowing at all what they're looking for. So at every point, they've only changed their parameters just one variable at a time, and not multiple things at once. This is a very key thing for all your experiments you run. Make sure you've only changed one thing, because then you can figure out what one thing actually caused the change you see. So now they're going to run their 4 different experiments with the 2 compounds they're interested in, which is quinine, the one they're really interested in, and mefloquine, which is the one they want to make some comparisons to. So they run it and they find one protein. Only one protein shows any change in the melting point, which is quite extraordinary if you ask me. And it is purine nucleoside phosphorylase. It does not matter whatsoever what this does or what it is. The point is they found the protein it targets. And it doesn't work for everything, though. So this is one strange thing they saw, right? So they saw that the intact cell experiment validates quinine. So the intact cell and the lysate, so the whole cells and the exploded cells, both agree on what the target is for quinine, so this molecule, but they don't agree on what the target is for mefloquine, so this one. This sort of validates what they already had done, right? Which is pick the four experiments, because they found 3 more proteins that were stabilized in the cell assay for this compound. But they figured out through other experiments and quite complicated literature search or whatever, but they figured out that these 3 other proteins are stabilized because of downstream effects. And these have to do with the cells responding to being treated with a drug. This has to do with the parasite basically being under stress by being given a drug and changing its expression of proteins and its various interactions with proteins as a response. So in the end, they're happy to conclude that this is possibly a target for both of them. And what do we see here is that they used multiple validations to find their target, which are the 4 different experiments we talked about. And they also used it to figure out that actually this is an artifact and that there aren't 4 targets for one of them and 1 for the other. Both, in fact, only target one thing. And what can you take away from this is that weird data can make sense, and it can also hold valuable information. So this will come into effect a bit later, you'll see. So now they don't want to just stop there. And again, this is, I think, where they're going the extra mile and why I really like this study, is they switch from monitoring what happens in the cell to taking this specific protein they found and characterizing it just on its own. So just the drug and the protein, not all the other stuff that's going on in the cell. Why is this important? Because it helps to decouple the protein from the pathways, and as we saw before, this seems to be a problem, at least with mefloquine, not necessarily with quinine. So they use, honestly, so I'm not going to go into every method they use now, even though they're fantastic, and I think it's excellent that they use so many different orthogonal methods, but you can basically teach a biochemistry course just from all the different things they did in this paper, so I'm not going to spend time going over every single method. But practically, they did something called differential scanning fluorimetry, which tells you again about binding and all these kinds of things. And they also used a 3rd compound, which they knew the value they're supposed to get, just to sort of validate again. They use something called surface plasmon resonance, which not only gives you binding, but it also tells you how quickly it happens. So this is why I say it gives you the kinetics as well of the binding. And it gives you also a number rather than just a plus minus or a temperature. So they got some numbers for inhibition on the protein. They did ITC, which is, long story short, it's just a titration where you measure how much heat is given out by binding. And this can also give you a binding constant, so a KD. And so binding constant is sort of just a number that expresses how strongly your drug binds to your target. The lower it is, the better. So 65 nanomolar is better binding than 11 micromolar, for example, right? And again, as I said, I'm not going to go too much into each of these, but what they're showing at each step here is that, yes, it works in the cell, but also there's protein on its own. You have all these different methods that still show you binding. So they're just, you know, hammering home their result and checking that it's actually correct what they found in their other methods. They did an enzymatic assay since this happens to be an enzyme, so they just tried to see if the enzyme stops working if you add the drug. And indeed, you do see that. And here they've sort of addressed another implicit assumption that they've made. So here they've assumed that because the drug binds, it also stops the enzyme working. This is usually a safe assumption, and it happens to be a safe assumption here, as they've shown. But it's always a good idea to show things that, you know, just make sense. And just because you think that's the case doesn't always mean it's true. And we have an example of that coming up in the next few slides, actually. So what's the key point here? Binding infers inhibition, as I just talked about, and they actually showed us. They didn't just stop at the inference. They said, okay, it's supposed to do that, but does it actually? And they show it. And then earlier you will have seen, yeah, thanks FW, yeah, that's quite helpful. Earlier we saw that they got something called a minimum threshold dose, which similarly to how binding infers inhibition, this minimum threshold dose can be translated to a binding constant or an IC50. But again, they don't assume that this is the case. They don't assume that the numbers are right. They go and they measure them and they actually see that they're quite similar. And what can you take away from this? So inference, so something that should work as it is always a good thing. You should use this, no question. But if you can, you should cross-validate with multiple methods and make sure that the results you're getting are consistent across different different setups and different experimental designs. So they keep going. They have gone way past what most papers would do to figure this stuff out, right? If you look at the two structures, these look quite similar, right? So you can see these two rings here, this preparidine ring here, this bridge with the alcohol. So doesn't matter the specifics of it, but I think everyone can agree that these are quite similar structures. They both target the same protein, and they look similar. So, you know, what everybody sort of assumed in the literature, and people were relatively happy to go along with, is that since they look similar and they target the same protein, they must bind in a similar way. But no, it turns out they went and they crystallized the entire protein with each of these compounds, and they looked at how they bind. Turns out they don't bind in the same way whatsoever. So the big yellow stuff here is the protein, and in this case they just have it as blue just so you can differentiate, but it's the exact same thing, so in both cases it's the same protein. And here in pink you have quinine, and in green here you have methylquin. So honestly, you can teach so much about medchem and structure-activity relationship, binding, and how all these things can change just by these two crystal structures. It's absolutely fantastic. Sorry, I'm kind of nerding up over this paper, but it's amazing. Yes, they really did do that too. They really went the extra mile. This is why I love this paper. This is one of my favorite papers I've ever read. So they went and they crystallized it, and they figured out that if you look at this, so this ring that contains the nitrogen here, right? In the pink structure, which is quinine, sorry, it's pointing underneath this aspartate here. Yes, it's in my second slide, so I can link the paper afterwards. Don't worry about that. So here you'll see that this is pointing in this direction here, and it's the ring on the left. And here, in fact, you'll see the ring is completely flipped. It's on the other side. It's not interacting here with this basic nitrogen is interacting with the aspartate here, but here it's not. It's interacting with a completely different residue. So honestly, here I could talk for hours on how this implicit assumption that medicinal chemists often make, that, oh, it looks similar, it binds the same, or it binds the same pocket, binds the same orientation, so SAR should be like this. This is very often nonsense. There's so many beautiful examples, and this is a perfect one, where even though they look so similar, they bind in a completely different way. And they also have different effects, as you saw, that they have different protein interactions with the cells, they have different binding constants. It's really not the same thing. But anyway, what do we take away from this is you can figure out why they bind differently. So they have completely different numbers. This one is way better at binding than this one, even though they look extremely similar. And they figured out why. And what you can take away from this is things that make sense are not always right, as I just spent some time talking about. Everything you assume is fine just because it makes sense should technically be shown by some kind of experiment. So this is what I would like you to take away from this. Just because it makes sense doesn't mean it's true. So some quick conclusions here. So they have very robust evidence, in my opinion, showing that this protein is the target of quinine. And now we know more or less how quinine works. So something that's been used for upwards of 400 years to treat malaria has only recently been characterized in how it actually works in the human body and against a certain parasite. Despite all this, I want to just read the passage from the conclusions of the paper, just to illustrate something. So they say, however, we cannot rule out the possibility that another therapeutic target of quinine exists in this or other stages of the development of the parasite, basically. And despite all these experiments they have and all this validation they have, they're extremely specific about exactly what their work can conclude. So they say that this is the most prominent target of quinine in the mid-stage of the developmental cycle. So this is all they conclude. They say that this drug probably works by targeting this protein in this part of the life cycle of this particular parasite. What? All right, anyway, I'm just going to ignore that. So the authors recognize the exact limitations of their work and how far it can carry them, and they make very precise conclusions about what they've done. And what I want you to take away from this is to ask yourself if your conclusion is precise and you've shown exactly what you've said. Have you actually conclusively shown what you claim? And here I've just summarized the takeaways from each of the slides that I've shown at the end of every slide, so you can read those later. Some summary of all this. And yeah, I think that's the end, and I'm open to take questions. It's only half an hour. Okay, that was a lot faster than I thought. I'm not going to record the questions just because, yeah, the file gets too huge then.